Hello, and thank you for joining us. I am Jasmine Navarez, Digital Content Coordinator at Simulations Plus. In this webinar, Dr. Chloe Brackus, Application Manager, will show how to develop MBMA models in Monolix from summary level data to support drug development decisions.
Thank you, and thanks for joining us today. Before I get started, I just want to also acknowledge the other team members who worked with me on this project. So Amit, Amparo, Geraldine, Heather, and Yassine. And also some of them are here today to answer your questions in the Q&A. So please don't hesitate to put any questions that you have that come up during the presentation. So today I'll first give an overview of MBMA concepts for anyone who's not already familiar with that, and then we'll go into two case studies. The first one is a bit simpler and more straightforward on naproxen versus placebo and osteoarthritis, and then we'll be able to do a more complex example with canakinumab versus existing treatments in rheumatoid arthritis. So first, I'm going to start by giving a bit more information about MBMA. So what exactly is model-based meta-analysis? So it's really a tool for integrating prior knowledge, and this can be used for decision-making. What's important to emphasize is that we're using summary level data. So this would be things like means over studies treatment arms, but with a full mathematical model. So we're not just looking at a single endpoint, but we can actually have a mathematical model to look at things like efficacy, safety, PK, PD. And so this means that we're able to integrate all available information, either from prior publications or internal data, because we're using that averaged summary level data. But because we have a mathematical model, we can actually take into account the fact that the trials might not be exactly identically designed, and we can handle that in the model. So that gives us a lot more flexibility. So what are some things that we can, applications of MBMA? So it allows us a way of comparing different drugs, either already on the market or being developed, that were not necessarily compared in head-to-head trials. So it's going to be a really good way of getting an idea of the current landscape and how that compares to a drug currently under development, and being able to use that information to evaluate decision criteria. And as part of that, we can actually use an MBMA to simulate clinical trials and get an idea of predicting trial success. And another thing we can use MBMA for is to look at surrogate clinical endpoints. So if we have existing published trials over a longer term, and say we might want to try to have something that would give an indication a bit sooner in time, an MBMA could be a way of evaluating the reliability of those surrogate endpoints. So comparing an MBMA to more classical meta-analysis, in a classical meta-analysis, often we're summarizing a single endpoint over several randomized controlled trials, whereas with an MBMA, we can really look at the entire profile. So we can say, look at efficacy, not just at a single endpoint, but also time to effect and duration of effect. And the totality of the data is really this advantage. So because we have the entire time course, this would be the difference of looking at, say, the entire survival curve compared to just a hazard ratio or an odds ratio for survival. And because we actually have a mathematical model, this is how we can really account for that heterogeneity in the design. Whereas with a more classical meta-analysis, we're often more limited to looking at trials with the same design. So continuing with the concepts, if we're looking at how an MBMA based on literature compares to a more traditional population analysis based on a clinical trial, our clinical trial, we're looking at individual patient data, whereas with our MBMA, we have our data averaged over multiple individuals within an arm. So we're looking at those aggregate results. In both cases, we're going to be using a mixed effects model as well as a structural model. But whereas in our individual patient data case, we have inter-individual variability for our random effects, in the MBMA case, we're going to have between study variability. And in the analysis of a clinical trial, we can optionally have inter-occasion variability, and that's going to correspond to between treatment arm variability. Finally, in both cases, we have residual error, but this is a little bit different because in the case of our MBMA, remember, we're averaging our individuals over an arm, and so the number of individuals is going to have an effect of how noisy that average is. So let's go into a little bit more detail about these different sources of variability. So first we have the between study variability. This is variability that's coming from the design, the inclusion criteria, maybe the equipment used for the study. So this is going to vary from study to study, and that means that we're going to have patient groups with different mean characteristics from study to study, but it's independent of the number of individuals recruited. So even if we recruit more individuals because of differences in, say, either the equipment measuring our samples or the inclusion criteria, we would still see the same between study variability. The between treatment arm variability, on the other hand, is based on the fact that when we recruit patients, we are randomly splitting them into 2 or more arms. And so the characteristics of these arms are not identical, but in this case, we do expect those characteristics to be more similar the more individuals that we have per arm. So we would be expecting recruiting more individuals to reduce our between treatment arm variability. Finally, we have the residual error, and this is representing the variation over time for the same arm, so the same group of individuals. And again, this is something that, because this is an average, we expect there to be less variability. It'll be less noisy over time if we have more individuals per arm. And so in the last 2 cases, because we think that our between treatment arm variability and our residual error are going to be smaller if we have more individuals per arm, we're going to take this into account by weighting that variability by either the number of individuals per arm or, if available, the standard error. So next, let's look at a bit more of what these models actually look like. So first, I'm going to start with just a standard hierarchical model. So this is for individual patient data. So this is probably familiar to most people here. We just have an observation for an individual at time j, and we are going to try to predict that observation with our structural model, which is going to be based on parameters per individual I, and then we also have residual error. If we look at our model for the individual parameters, for each parameter for each individual, we have the typical parameter that we're estimating for the whole population. We have any covariates that we're including in our model, so that are going to be differing per individual. And then we have random effects for our inter-individual variability, or basically how much our individuals differ from that typical population parameter value. So with this model, we basically have the components of the structural model, the residual error model, and then we have distributions for our parameters, distributions for our observations, and the covariate effects. We have 2 levels of variability, the inter-individual variability and the residual variability. We can then add occasions to the standard model. So now I have my observation from an individual I at occasion k and time j. So this means we can have parameters that vary not just per individual, but also per occasion. And that means that when we are looking at our model for the individual parameters, they vary by occasion. And so our covariates can also vary by individual and occasion. And we now have 2 random effects. We have both the inter-individual variability and the inter-occasion variability. So the components of our model are all the same, but I now have 3 sources of variability because I've added that inter-occasion variability. So now we can move on to how we're going to implement our model in the case of MBMA. And I'm not going to get into a full mathematical derivation here, but just know that you can actually derive in the kind of more straightforward case, we can sum up an individual patient data model, and then we're going to get the averages over arms. And so we can actually derive the model that we use in the case of MBMA from models of individual patient data. And so here I have my observation, which is for my study, my treatment arm, and at my time j. And so again, I'm having that made up of my structural model and my residual error model and my structural model, I have my parameters that can vary per study and per treatment arm. You'll notice that now my residual error, I have my weighting, right, where I'm weighting my residual error by the number of individuals that I have in my treatment arm. And so now if we look at the individual parameters, I have my typical parameter for the entire population. And then I can also have covariates, which can vary per study and arm. But that means I need to have my covariates. I can't have, say, the weights of individual patients, but I would need to have the mean weight over my treatment arm. But if those differ a lot from, say, study to study, I can still take that into account in my model. And now for my random effects, I have my between study variability and my between treatment arm variability. And you can see that my between treatment arm variability is also weighted by the number of individuals. So let's look in a bit more detail how we do this weighting in the case of MBMA. So we have, we want to weight both the residual error and the between treatment arm variability. And there's two different ways we can do this. We can weight by the number of individuals per arm. And so this is per study, per arm, and also it can be per time point because we could have dropouts. So we could have the number of individuals per an arm changing through time. And so here you can see that we have both the residual error weighted by the number of individuals. So it's going to be smaller the more individuals we have in our arm. Our between study variability is unweighted, and the between treatment arm variability is also weighted by the number of individuals per arm and will be smaller with the more individuals per arm. What we can also use, if it's available, is the standard error. So recall the standard error is just the standard deviation divided by the square root of the sample size n. And so if it's available, this is good to use because this way we avoid assuming that the standard deviation is the same across studies. You can see in the top where I'm weighting by the number of individuals, I just have a single, say, sigma squared parameter here that I'm fitting for my residual error. But if I'm using my standard error, those can also vary per study, per treatment arm, and per time point. And so I don't have that stronger assumption that those are always the same. So I can use the standard errors for the residual error. Again, the between study variability is unweighted, and for the between treatment arm variability, I would, like in the first case, I would again just weight by the number of individuals per arm. So if we are doing this weighting in both our residual error and our random effects term, we need to actually handle this in Monolix. Random variables need to have a constant variance. So we're going to need to do a model transformation so that we don't have our error depending on non-constant parameters. And here we know for weighting by the number of individuals that this is changing per study per arm and also possibly by time. So how we can do this, we can rewrite our same model equation, and we can bring our number of individuals per treatment arm outside of the random variable. This way we have a random variable that's just mean-centered with a constant variance. And now we can just multiply both sides by the square root of n, and this gives us our transformation. So here we're going to transform our observed data in the data set by multiplying by the number of individuals per arm, and we're going to do the same transformation to the model prediction in the structural model so that we're comparing the same thing. Now, if we're instead weighting by the standard error instead of the number of individuals, this looks really similar. So here I have my residual error term using the standard error, and I can again move that outside, divide both sides, and that gives me again my transformation that I'm going to apply in the data set and in the structural model. And note that we can fix instead of estimating a constant variance for our residual error, we can fix that to 1 so that we're explicitly using those standard errors that we have from our data. So how does this look like in the data? So basically in my data set, I have my observation column Y. I have another column for my N arm, and I'm going to add a column to my data set where I apply this transformation of multiplying by the square root of the number of individuals per arm. And then I'm going to do the exact same transformation in my structural model. So I can read in the number of individuals per arm as a regressor. Then I can calculate my effect And after I calculate my effect, I apply this exact same transformation, which I use as my output. And that way I'm comparing my model predictions and my observations on the same scale. So this is basically a quick introduction, just conceptually. If you have any questions on that, don't hesitate to put them in the chat. And now we're going to go into our first case study. And so this is our case study looking at naproxen, which is a non-steroidal anti-inflammatory drug used to treat osteoarthritis, which is a wearing out of the protective cartilage. And this data set comes from the paper Boucher and Bennett's, which is a really nice tutorial paper where they implemented this model and compared it with bugs, non-mem, and NMLE. So we're going to implement the same model in Monolix. And what we have as our observed value is the WOMAC pain score. This is scaled to be a value between 0 and 10. And the studies have either a flare or a non-flare design. A flare design means that there was a washout of existing pain medication and a certain criteria for how much the pain increased, so basically a flare-up. And the question that we're interested in answering are two. First of all, we want to compare the onset of action and maximal effect between treatments, so between naproxen and placebo. And then secondly, we want to examine the impact of the flare design on the treatment effect. So before we get right into the modeling, we're just going to I'll just do a quick look at the funnel plot for publication bias. So here what we have is we have the standard error on the y-axis and the treatment effect, so naproxen minus placebo on the x-axis. And we have that because we have longitudinal data, we have that for different time points. We can see that the mean treatment effect is here, this vertical line, and then we kind of have implicit confidence intervals based on the standard error. And what we want to see is we want to see basically all of our data kind of inside this funnel. The idea being that larger studies with smaller standard error are going to give better estimations of this treatment effect, and that we don't want to see a bias, the idea of the file drawer effect, where small studies that didn't show an effect were not published or not used, and only those with the large treatment effect were used. So we basically do want to look for asymmetries in this, and we kind of want all of our data roughly randomly distributed within our funnel. So here we don't see any evidence of publication bias. We can go ahead and move forward with our analysis. So for our data set, we have study treatment arm. We have whether the treatment was placebo or naproxen. We have both the number of individuals and the standard error, which I'll get to. We have flare. So this is 0 for a non-flare design or 1 for a flare design. WP is our observed response, so that's our WOMAC pain score, and the WPSE is our standard error of the pain score. And how do we get that standard error? So if we look at the different studies that we're using in our meta-analysis, so here's an example where they're reporting both at different time points, the mean for here placebo in gray, and this is 2 different treatments, as well as you can see these error bars are the standard deviations. And so we can basically take the reported standard deviations and the number of individuals per arm for the study to calculate our standard error. If we then look at our data, you can see that we have groups for both naproxen and placebo, and within each of those we have our studies that were the flare design and the non-flare design. And we can see that we basically have our pain starting from a higher, basically the flare level for the flare design, and then with treatment decreasing to a plateau. And so for our model, we're going to look at 3 different parts. First is the structural model. So how do we want to model that response, our WOMAC pain score? Then the residual error model. So how are we going to weight that by the standard error? And then finally, for the parameters in our structural model, how are we going to model those parameters? So let's start with the structural model. Here we have an Emacs model. So just a really simple model. We're going to be starting from an initial E0. We model our effect as decreasing until a saturation point, which is Emax. And we have our ET50 parameter that we're modeling, which is our time to 50% efficacy. And you notice here we don't have any kind of drug concentration. We're really just modeling this with over time. And you can see here, this is what the structural model would look like for defining the same model. So next, let's look at our residual error. So here our WOMAC scores are on a scale from 0 to 10, but for simplicity, we're just going to assume that these are normally distributed. In our case, the scores are all in the middle of the range, so this is a reasonable assumption, but of course we could use some kind of logit model or something else if we wanted to make it more complicated. And we think that the noise of that is going to be smaller when the standard error is smaller. So here, oops, sorry, I went back too far. So we think that the standard error we're going to use to weight our residual error. And so you can see here, I have my observation, my prediction is my structural model, and then my error is using the standard error. So in my structural model, I have my 3 parameters, E0, Emax, and ET50. And so next, I'm going to look at how we're going to structure each of those parameters. So first for E0, we're again just going to treat it as normally distributed. We do think that E0 is going to depend on the study design. If you remember, we saw a higher E0 for the flare design compared to the non-flare design, so we'll include that as a covariate. And we're also going to include between study variability, and that's the variability that's not weighted on E0. And just for simplicity, in this first case, we won't include between treatment arm variability. So our model for E0 is we have an overall population parameter estimate, a covariate for whether the design is flare or non-flare, and then a random effect for the between study variability. Next, we can look at the parameter Emax. We're again going to just treat this as normally distributed. For Emax, this could depend on the drug, so we expect to see different treatment effects for naproxen and placebo, as well as the study design, right? We might have a difference in how much of a treatment effect we see, depending on if it was a flare or a non-flare design. So we're going to include both of those as covariates, and then we'll also include between study variability as our random effect, and for simplicity, not the between treatment arm variability. Lastly, we have our parameter ET50. ET50, this is the time until 50% efficacy, so this needs to be positive, so we'll use a log normal distribution. We do think that this could depend on the drug, so we'll include drug as a covariate. And for simplicity, we're not going to include between study variability or between treatment arm variability. So to summarize, we have an Emax model with 3 parameters, E0, Emax, and ET50. Both E0 and Emax are normally distributed, and ET50 is log normally distributed. And we have a treatment effect on Emax and ET50 and a flare effect on E0 and Emax. And we have between study variability on E0 and Emax. And for our residual error, we are weighting by the standard errors. So next we need to look at what kind of model transformations we need to do in order to implement this model. So first for the residual error model, so again, we need to have our error term defined with constant variance. So we're going to do a transformation. So we'll bring our standard error outside of the random variable. That way we can divide both sides by that. And that gives us our transformation. So we need to transform both our observed data by dividing by the standard error, and in our structural model, we'll need to divide our prediction by the standard error. And so let's look at what that looks like. So here we have our observed data. So we have our WP, our pain score, and we have our WP-SE, the standard error. And we make a new column that's going to be the transformed observation, where we just take the WP column and divide it by the WP-SE, and we put that as what we can use for our observation. And in the model prediction, we're going to read our WP-SE column in as a regressor. That way we can use it in our structural model. We're going to calculate our model prediction here, F, and then we'll make a transformed F where we just do the exact same thing. We also divide by the standard error, just like we did for our data, and we'll use that in our output. Now, next we need to do the parameter distributions. Now, in this case, we don't have any weighting, so normally we would be able to just include this in the GUI. But because of some technical reasons for how occasions and covariates are handled in Monolix, we actually do need to do this in a structural model. But this is going to be good practice for in the next example when we'll also be including the between treatment arm variability. So the way that we're going to do this is we're going to define two variables. So we have Emax. This is for our fixed effects. And then we have this eta BSV Emax, and this is going to be our random effects. And so these are both just going to be normally distributed. And so that means inside of our structural model, we can just add them together, and that's going to give us our Emax with random effects. And so that means if we now look at our full model, after we've calculated our Emax with random effects, we can just put it into our Emax model here. Then we do our transformation that we talked about so that we can match our model prediction to our model of data and use that as our output. So this is everything we need to do to know how to set up this model. So now let's switch to Monolix and look at how we would actually implement this in Monolix. So what I'm going to do here is I'm just going to do a new project. I'm going to browse to my data file and open up and just wait. I'm sharing my screen, so it always takes a little bit longer. And so now I'm at the point where I need to tag my different columns. So here's my study column. This I'm going to tag as ID, because remember the different studies are analogous to my individuals if I'm modeling a single clinical trial. Here's my arm, and this is going to be my occasion variable. I have my treatment here as categorical covariate time. Here I have n individuals. If I wanted to, I could also tag this as a regressor if I was going to be including between treatment arm variability. But in this case, I don't need it, so I'll just leave it ignored. I have flare, which is going to be my categorical covariate I can use to include my flare effect. And here's my WP. So this is my observation, but this is my untransformed observation. So I'm going to leave this ignored. And here I'm going to tag my transformed observation as observation. And then the other thing I'm going to tag here is my WPSE. I'm going to tag this as regressor so that I can use it in my structural model. And then I have a couple other categorical covariates that we'll get to later. So this I'm going to accept. And now I'm ready to, I can browse to the model file so that we already looked at. And just wait a second for it to load. And so here's the structural model that we just looked at where I have my Emax with random effects. I calculate my Emax model and I do my transformation to divide by the standard error. And I have mapped in my data my transformed to my transformed model prediction. So that's my structural model. We can now switch to the statistical model to set that up. So first for my observation model, I want to pick a constant error model, and I again want a normal distribution for this. Now if I look at my individual model, so this is for my model for my parameters, for E0, right, we said that was, we're going to model that as a normal random variable, and same thing as Emax, we're just going to model as normal. For ET50, this needs to be positive, so I'm going to keep this as long normal. And then this is my random effect, so this I'm also going to pick as normal. For random effects, I want to include random effects for E0, and this is on the ID or study level. So this is my between study variability on E0. For Emax, right, I've defined this manually here. So I'm going to remove it here because I'm handling the random effects with this variable here. And then for ET50, we said for simplicity, we won't include random effects. And for Emax, this is my between study variability. So that means I need my random effects here at the ID or study level. If I was going to include between treatment arm variability, that would be when I would want to add random effects on the arm level. So that is how I want to set up the random effects in my model. The next thing that I can do is add the covariates. So for flare, I have 0 for a non-flare and 1 for a flare. So I can just use this as is, and the non-flare design will be my reference. For the treatment, I'm going to add a covariate so that I can go ahead and allocate these. And this way I can make my placebo the reference category. So now I'm ready to add my covariates. And so we said that we wanted to have our treatment on Emax and ET50. So I'm going to use my transform treatment here that I just added, and I'm going to add that to Emax and ET50. And then for the flare design, that we wanted to have on E0 and Emax. So I'm going to add that there. So now I've set up my statistical model, and the next thing I need to do is set my initial values. And so here, I can use the auto init, but I can also go ahead and just set these based on my data. So if we go back to the slides and I go back to looking at what my data is, I can use these values to come up with good guesses for what my initial values should be. So first of all, I have E0. And so E0, this is going to be for my placebo non-flare at time 0, what is this value starting at? So I can say that looks like about 5. So I can put 5 in for here. For Emax, this is going to be how much of a treatment effect I see for my placebo non-flare. So I could say that looks like about 1. So I can leave this as 1. And then for ET50, this is going to be the time to 50% effect. And so I can look here, and so this here, my time is in weeks, and this is time 0, and here's my data points at time equals 1. So it kind of looks like I already get in less than a week, basically in 1 week, I already have more than 50% of my effect. So I can pick something like 0.5 here. This parameter here is my random effect. So here I want, this is my normally distributed random variable with a mean 0. So here I want to set this to 0, and I also want to fix this because I don't want to estimate this. I only want to estimate my random effects for this variable. For my random effects, I can leave the initial values both at 1. And so now I need to look at my covariates. So the first thing is on E0, my covariate for the flare design. And so for this, I need to look at the difference between placebo flare and non-flare at time equals 0. And I could maybe say this looks like going from 5 to, say, 7. So I could just make this, let's just say 2. Here, this is the effect of flare on Emax. So this is basically for placebo, how much of a difference I see of a treatment effect. And so I could say maybe this looks like, and this is additional, right? So if I said this is 1, I could say that this is going to be, say, one more. So I'll make this a 1. Then I need to say the effect on Emax of my naproxen. So this is basically looking at the difference from naproxen non-flare and placebo non-flare. So I could again say maybe 1. This is like 1 more, looks good. And then finally for my ET50, I need to look at this time of getting 50% efficacy. And I could say that this looks a little bit steeper to me than this. So I'll add a small positive value. Let's just say 0.2. And the other thing I can do is I can fix my residual error parameter. I can fix this to make sure that I'm only using the standard error in my estimation. We can go ahead and look at check initial estimates to make sure that these values look reasonable. And then now I'm at the point where I'm ready to save this and run my model. And so just in the interest of time, it doesn't take that long to run, but it does take a little bit. So I'm just going to switch here to one where I've already run my model. And so this is the exact same setup. And now I have plots. So for example, I have my individual fits that I can look at. I also have observations versus predictions. So I have all of the standard diagnostic plots. I'm not going to go into this in detail because it really is kind of the same as doing just an individual patient data, but basically all the same plots are available to me. Something I do want to do is go ahead and look at the assessment. So before I start doing too much, say starting to look at covariate effects or things like that, where I'm going to be comparing models based on the likelihood, I want to make sure that my likelihood is actually stable and my parameter estimates are stable. So for that, I can go to assessment. For people who haven't seen this before, here I can pick how many runs I want to do for my convergence assessment. Each one will be started with a new random seed. And I can also, if I want, vary the initial conditions. So it'll vary them a little bit around where I had set my initial values. And so that way I can make sure that just starting my estimation from slightly different initial values doesn't wind up in a very different set of estimated parameters. And so if I look at this, I can see that most of my parameters are quite well estimated. For example, my treatment effects on both ET50 and Emax. I can see that there is more variability in both Emax and my covariate on flare. So this basically, and these look like they might be a bit correlated. So basically it looks like there's a little bit of identifiability issue, that the model is having a hard time partitioning the treatment effect for placebo between the flare and non-flare designs. But if I actually look here, I can see my likelihood. And so the scale is going, this is 486.1 to 486.7. So these likelihood values are differing by less than 1. So basically my model likelihood is very stable. Just with I can get the exact same likelihood by slightly changing a little bit some of my Emax and the flare covariate on Emax. So this looks good. I can kind of proceed to looking at my model. So we can go back to estimation and look at my estimated parameters. And I can see also that all my parameters are well estimated. My RSEs are all good. So let's go back to our question and see how we can answer our questions with these model results. And so our first question was, how does the onset of action and the maximal effect compare between treatments? And so we can answer that first. For the maximal effect, we can look at our covariate of naproxen on Emax, and we can see that naproxen lowers the pain scores by about 0.8 of a point more than placebo. And then next, for the onset of action, here, because this is a log normal parameter, we need to transform it by exponentiating it. And then if you remember, our time is on weeks, so we can also multiply it by 7 to switch to days, and we can see that naproxen reaches 50% efficacy 2.2 days sooner than placebo. Something else that I'll point out is that there is a really handy setting where you can get fixed effects by category in the results tables. And so if we go back to Monolix, basically that's just the setting here. If I turn this on, I get this new section. So here's the fixed effects we already saw, which will give me my covariate values versus the reference value. And this will calculate them by category. So now I have, for example, my E0 for non-flare and flare, and I have my Emax for all my different combinations of flare and treatment. So this can be really handy that I don't have to calculate these manually. So now let's go back to our question 2, which was, what is the impact of a flare design on the treatment effect? So if you remember our Emax parameter, we modeled as having a population parameter estimate, and then we have our 2 different covariate effects for treatment and flare. But if you remember, we're basically treating them here as independent. So that means we're not considering any potential increase in Emax for naproxen with a flare design compared to what difference we already see between flare and non-flare design for placebo. So we don't see any extra, for example, increasing Emax for naproxen with the flare design. So if we want to answer this question, we need to add a covariate for the interactions, basically the interaction between treatment and flare. And so to do this, we can basically have a column in our data set that defines that interaction of treatment and flare. So this column is going to be 1 only in the case where we have an naproxen and a flare design. So you can see here, placebo flare, it's 0, naproxen and flare, it's 1, but placebo non-flare 0, naproxen non-flare 0. So now we can use this as a covariate on Emacs. And so this, again, I already have a project with this. And so you can see here, I have set up, we have that interaction of treatment and flare covariate. And so I've added this as a covariate on Emacs. And if we go and look at the results, I can see that that parameter is estimated very close to 0, so a very high RSE. And if I also go and look at tests, I can use the Wald test to test whether a covariate should be removed from a model. And so here I can see it does look like this covariate is non-explanatory and should be removed from the model. And so what that means is my answer to question 2 is that there is no impact of a flare design on the treatment effect. So basically we don't see any increase in Emacs by having a flare design with naproxen over what we already see with the placebo. Next, I can just show you really quickly. So the Boucher and Bennett paper, again, was the tutorial paper where they implemented the same model in bugs, non-mem, and NLME. So here is just showing that also with Monolix, we get very similar parameter estimates. Here I've slightly, I've run the model just reparameterized to match the parameterization in the paper where they estimated, for example, the log of AT50 directly. And you can see the parameter estimates are all very similar. We do have a slightly higher estimate for Emacs. But this is really, if you remember, this is where the parameter we saw that there was more variability in the assessment. We can also end up with similar values as the paper with just slightly different initial values. And using the same, and we end up with the same likelihood. These are just the ones I get when I kind of use my fairly round initial values by looking at the data. So in conclusion from this case study, we say that naproxen lowers the time of onset and increases the maximal effect in osteoarthritis compared to placebo. We don't see any additional effect for naproxen with a flare design, but MBMA allows us to really easily integrate information across numerous clinical trials, both published and unpublished. And so now we're going to move on to our second case study. This one is looking at canakinumab versus existing treatments in rheumatoid arthritis. So this is a treatment that was developed for patients that were already treated with an existing treatment. We have data from a phase 2 trial. And this example is based on a paper Demon et al. And so we're using basically the same studies from that paper with similar inclusion criteria, as well as a few more recent studies since that paper came out. And here our response variable of interest is ACR20. So this ACR stands for American College of Rheumatology. And the 20 means that we're looking at the percentage of individuals with at least a 20% improvement of symptoms. And our question here is, we have the drug canakinumab, which is under development, and we want to look at the efficacy based on our what we have so far from our phase 2 trial compared to 2 other drugs that are already on the market. We can look at a funnel plot for publication bias. Here, we don't really have enough studies to make this kind of fully valid. In this case, we don't have standard error. So on the y-axis, we're looking at 1 over the square root of study size, remembering that the study size is really one of the main determinants of that standard error. And We're also grouping our different drugs, so right, which could have different treatment effects. So the fact that we kind of see more red on one side, that's not really the asymmetry we're worried about. We can notice that in this case, we do still see kind of a funnel shape, but we don't really have enough data. We really need about roughly 10 studies to really be able to use a funnel plot, but we can at least look at this as there's nothing jarring that jumps out at us for publication bias before we move on. If we look at our data set, we have here, again, the study, the arm, the drug. Here we have our ACR20 is our response. We have an arm for the number of individuals per arm. And then we have several different covariates that we'll be able to look at later to see if they have any effect in our model. Looking at our data, so here we have 3 different drugs, and the canakinumab that we're kind of currently investigating is here on the right in green, and in all 3 cases we have the placebo plotted in black. And so what we want to do is try to model this response we see of kind of this increase and plateau. And again, this is the percentage of individuals with a 20% improvement. So for our modeling steps, we're going to go through the same 3 parts of looking at a structural model for the ACR20 response. For our error model, this time we're going to be using the number of individuals per arm to do the weighting, and then also looking at the parameter distributions for our parameters in the structural model. So we'll again use an Emax model. This time our prediction is increasing to saturation. So we have 2 parameters. Emax, the maximum effect, and then our T50 for the time to 50% effect. And our model looks really similar to what we already looked at. The model for our residual error, now we're going to get a little bit more complicated. So we have our ACR20 is a percentage. We're going to model it between 0 and 1. So that means we need to ensure that it stays between 0 and 1. And so we're going to use a logit transformation, and we want to apply the error to that for both the data and the prediction. And now we don't have standard errors available. So instead, we're going to use the number of individuals per arm. And we want to use that to weight our error because we think that the residual error should be smaller when we have more individuals. So here we have our logit transformation applied both to the observation and to the prediction. And then that is what we're going to actually apply our error to so that this can be our normally distributed error. So our 2 model parameters here, in this case for the prediction, is Emax and T50. So let's look at how we're going to handle those parameters. So for our parameter Emax also needs to stay between 0 and 1. So we're also going to use a logit transformation for it. Emax we think could depend on the drug, so we'll use drug as a covariate. And here we're going to use both between study variability, which is not weighted, and between treatment arm variability. So our model for the Emax parameter is that we take a logit transformation that ensures it'll be between 0 and 1. So we also take a logit transformation of our population parameter. We add a drug covariate so that we can have an effect on Emax of the drug. And then we have 2 random effects for both the between study variability unweighted and the between treatment arm variability, which we're going to be weighted by the number of individuals per arm. Then we'll have our parameter T50. This needs to be positive, so we'll use a log normal distribution. We also think it might depend on the drug, so we'll include drug as a covariate. And just for simplicity here, we're not going to include between study variability or between treatment arm variability, although that certainly could be included in a more complicated model. So to summarize our model, we have an Emax model with a logit transformation to assure that our observation stays between 0 and 1. For our 2 parameters, we have Emax, also with a logit distribution, and our T50, a log normal. For both of them, we have a drug covariate. And for Emax, we have both between study variability and between treatment arm variability. And if we look at our our residual error and our between treatment arm variability, those are both weighted by the number of individuals. So we're going to need to do some model transformation in order to be able to do that weighting. Again, because we need constant variance. And so this looks a lot like what we did before. It's just a little bit more complicated because we actually have this logit transformation. So we have the logit transformation first. We again multiply both sides by the square root of n, and that gives us our transformation. So we have here our transformation we're going to apply to the observed data. So this is in our data file. We're going to make a new column, and the transformation that we do, the same transformation to our model prediction. So let's take a look at that. In our data, you can see we have our column Y, we have our column N-arm, and then we're going to add this new column, the transformation of Y, where we take the logit of Y and then multiply by the square root of N. And we're going to do that exact same transformation in our structural model. We can read in the N-arm column as a regressor. That way we can calculate our prediction for ACR 20, and then to that prediction we apply the same transformation, and we use that as our output so that we are comparing the same thing. So that's the error model. We also need to worry about the model transformation for the parameter. So if you remember, for our Emax parameter, here is where we're weighting, and we have our random effects due to the between treatment arm variability, which is where we wanted to weight by the number of individuals per arm, because we think that that between treatment arm variability is going to decrease with more individuals per arm. And so we do the exact same thing. We can move that square root of n outside of our random variable. This leaves us with a random variable with constant variance so that we can estimate in Monolix. And if we look in the structural model, we can see how we actually are going to implement that weighting. So here in our input, we have 3 parameters. So we're basically decomposing our Emax into the fixed effects, and then our 2 random effects for our between study variability and our between treatment arm variability. Now, what we need to do in the structural model is a little bit more complicated than last time because we have this logit transformation. So if you remember, we said we wanted our Emax parameter to be between 0 and 1. So we're going to use a logit normal distribution for that. So that means in order to be able to add our random effects, we need to take the logit transformation, because this is going to be a number between 0 and 1. We need to get it onto something that's going to be on a normally distributed scale. So now with the transformed, we can simply add on our random effects. And this is where we handle the weighting. So right here, our between study variability is unweighted. Our between treatment arm variability is weighted, and so we divide by the square root of n arm that comes as a column in our data via a regressor. So after we've done that, we just need to do the back transformation this again, so that we have our Emax with random effects, and this will again have values between 0 and 1. And so now if we look, here's our full structural model. So here, this first part is what we just looked at, where we take our, we do a transformation for the fixed effects, we add the random effects, we transform back. So now we have our Emax RE. This is our Emax with random effects, is going to be between 0 and 1. So now here's where we have our Emax model, where we calculate our predicted ACR 20 using that Emax term in our T50. We have a saturation. This is just to avoid any numerical issues with taking the logit of 0. And finally, we need to transform our prediction. So this ARC20 is our prediction, and then now we're going to apply that same prediction that we applied in our data, where we take the logit and multiply it by the square root of n arm. So that's everything we have now set up. So now let's go and do that in Monolix so that we can see how to do the setup in Monolix. So I'm just going to make a new project, and we'll browse to our data file and just wait 1 second for it to load. So now we're going to do the tagging. Here, I want to tag first my study. This is going to be my ID, right? Because my study is analogous to an individual. My arm is again going to be occasion. this drug I'm going to use as a categorical covariate. I don't need the integer for it. I have time. And now here I get to y. This was automatically tagged for me as an observation, but I actually want to ignore it because I really want to tag my transformed y as my observation, right? Because this is my y that I've already done my transformation. Then I can go back here is my n-arm, and this is what I'm going to tag as a regressor to use in my structural model. And then I have my different covariates. I'm just going to tag all of these as continuous covariates, and we can evaluate them later to see if they are important or not. So I'm just going to accept that. And now I can browse and find my structural model. So if we go back and look, so this is the same structural model that we just saw with doing the different transformations. And then in the end here is I've done the same transformation to my model prediction as the data, and I have mapped my data transformed column to my transformed model prediction. So now we can go to statistical model and tasks to set up our statistical model. So for the observation, again, I'm going to change my error model to be constant with a normal distribution. And now I can do my individual model for my parameters. So first, for Emacs, I want to make this logit normal, but I'm first going to go back and just make the initial value just something between 0 and 1, so that I'll be able to properly set the boundaries. So now I'm going to make this logit normal, and this way I can check that the limits of my distribution are 0 and 1. And so I just didn't want to have my initial value as 1, because then that would not be allowed to be right on the boundary. For my T50, so this I'm going to keep log normal. This is just something that needs to be positive. And then these are my two random effects, right? So both of these are going to be normal. Next, we can look at how to set the random effects. So for Emacs, I am defining my random effects manually with these two variables, so I'm not going to define them through the UI. And for T50, we just, for simplicity, are not including them. So now I need to set my random effects for my two different variability, the between study variability and the between treatment arm variability. So for the between study variability, this is perfect. I want my variability on the study level, which is the ID level. For the between treatment arm variability here, I don't want it on the study level, but I want it on the treatment arm level. So this is my two different random effects that I want to have on Emacs. And so I have my between study variability on my ID study level and my between treatment arm variability on the arm level. So that is the random effects. Next, I can look at covariates. And so we want to add a drug covariate on both Emacs and T50. So I'm again going to add a new covariate so that I can make placebo my reference category. And then I just need to add this new drug on both Emacs and T50. So this is everything set up for my statistical model. We can now go to initial estimates. The first thing I'm going to do is fix my two random effects to have a mean of 0, because these are my normal random variables where I only want to estimate the variance or the random effects. And then we can go to check initial estimates. I can see that 0.5 looks good for Emacs. T50 looks like it might need to be a little bit bigger. I could try, say, something like 5. So this looks good. I can set these as my initial values, and now I'm ready to save and run my project. So I'm just going to switch to my project that I've already run. So here you can see everything is set up exactly the same. And so I can, again, I have, here's my individual fits, and I can have all of my different diagnostic plots, observations versus predictions, etc. Before I start looking too much into exactly how well this fit, I'm again going to look at the assessment to see if I have stability there. And so again, I'm going to be running 5 different runs with different random seeds and different initial conditions for my parameters at each run. And here I can see that this is actually more problematic. So here, my likelihood is varying from about 685 to 720. So that's 35 points. And I have a lot of variability. We can actually look here. You can see I have a lot of variability in my different parameter estimates. So this is problematic. We can see that the trajectories vary a lot from run to run. And something to know is that parameters without variability, so here this is Emax, because I'm defining my variability myself in the structural model, this means the SEM algorithm doesn't have this it's not the SEM algorithm that's kind of estimating that as part of this parameter. And also T50, I didn't include random effects on that. And so that makes it harder for those parameters to be estimated. So this is something to potentially look out for. What can we do about that? So we can change some of the SAM settings for how to optimize parameters without variability. The default is for parameters without variability is to just do optimization using the Nelson-Mead Simulx algorithm. Instead, we can choose to add artificial variability. So what this does is it adds artificial variability so that the SEM algorithm is able to estimate that parameter better, and it's progressively forced to 0 so that there's no variability left at the end of the estimation process. And there's also a possibility to do this in 2 different phases with artificial variability and then the Nelson-Mead algorithm. And which one works better really depends on the exact project and model. So it's really a case of just trying both of them. So I can switch to a project where I've done that. And so what this looks like in the interface is just on the population parameter tasks. I can look at the options. And what I've done here is added variability at the first stage. And so now if we actually go and look at the assessment, we can see here the difference in likelihood is less than 2. And if I look at my parameters, I can see that they're all much better estimated. So let's go back and actually look at the estimation. So I have, again, my individual fits, my observations versus predictions, etc. If I look at my results, I can see that I have some high RSEs for some of my beta covariate parameters here. In particular, it looks like it's having a hard time estimating the beta effects for some of my drug parameters on my T50 parameter. It looks like they're getting estimated maybe close to 0. I can also go ahead and look at tests. And so it's basically suggesting that these modalities in my covariate are maybe not different than the placebo. So what I can do to handle this is I can make a new covariate that basically groups my other drugs with placebo and is only going to be looking at adalimumab as separately as a different time of onset T50. Here I can also see it looks like my canalimumab looks like the Emax might differ than placebo. In this case, I do have individual patient data that did show that this was different than placebo. So for now, I'm going to leave this covariate and we'll see what happens if I take this into account. So how I would do this is I'm just going to go add another covariate, and I'll just call this grouped drug. And what I'm going to do is this is the one that I want to be different. And then the other 3, I want to all be considering the same as placebo. So I'll make that the reference category. And then what I can do is just switch for my T50, I'm going to use this group drug instead of the one with all 4 separately. And so if I go ahead and do that, you can see here, I've done the same thing. I just have my group drug covariate on T50 and my drug with all 4 separate on Emax. And so now I can go to results and I can see that everything is well estimated. Here I have infinity for my CV, but this is not concerning. This is because I've actually fixed the mean of both of these to 0, so I'm dividing by 0, so it's expected that I would have infinity here. I can go ahead and look at my tests, and I can see that, yes, I should keep all of these covariates in the model. So the question is kind of how would I interpret these random effects? So let's go back and look at the slides for that. So here's my random effects, right? I have my between study variability, my between treatment arm variability. So recall from the structural model that we just added both of these to the fixed effect for Emax, but this one is weighted. So in the first case, the between study variability is simple. I can just look at this and say, okay, it is 25%, and this is my variability from study to study of what percent of patients achieve that 20% improvement. For the between treatment arm variability, I need to remember that I weighted by the square root of the number of individuals per arm. So to interpret that, I need to take a typical N per arm of, let's say, 200, and divide by the square root of that so that I get my 14%, which is about the variability between arms of the maximal percent of patients achieving that ACR 20 response. So again, I'm not going to go into the details of the model diagnostics, but that proceeds really standardly. And so I can, from what when I look at all that, I don't have any model misspecification. So we can accept this model and move on to doing a covariate search. So I have several different covariates, the C-reactive protein, disease duration, swollen joint count, and I might wonder if any of these could also influence Emax. Here, I'm just going to take a simple backward approach of adding all the covariates to the model and then assessing them with a weld test. You could certainly do adding covariates one by one. You could use COSAC to evaluate. So this also proceeds really like a standard evaluation of covariates. So in my case, I decided to add all of my covariates at once, and we see here that none of these covariates are significant, so I'm not going to keep any of them in my model. So my final model is the one that we just saw. Something else that I'll point out here is because we had to transform our observation in the data file, that means that all of my different diagnostic plots are done on that transformed scale. But sometimes it is more intuitive and more interpretable to be able to look at these plots on my original scale. So this is also possible to do. We have our code available for download if you just go to the MonolixSuite documentation for the Lixoft connectors, the RAPI, and there's examples there. So there's a script you can download that will let you back transform kind of all the different model predictions and model results so that we can look at things like, say, individual fits and the VPC on our original scale of ACR 20 as a percentage here from 0 to 100. But going back to our actual question, could canakinumab be more effective than our other two treatments? So we can look at our estimated true efficacy over an infinitely large population. So here, again, I can get my Emax for each of my 3 treatments. I can either use that display fixed effects by category, and Monolix will calculate this for me directly in the results tables. I can also go back to my formulas and calculate this myself by using my Emax fixed effect estimated for my placebo and taking into account my different covariate values estimated for each of my drugs. But one thing to keep in mind is that these values are marred with uncertainty. So these are kind of my point estimates, and here I would say that canakinumab is not more effective, but this is just based on my particular clinical trials. So if we want to compare the true efficacy over an infinitely large population, we could perform a large number of simulations of these 3 different treatments and the placebo, and what we can do is draw that Emax value from its uncertainty distribution. So we don't want to just assume that we precisely, perfectly estimated it, but we want to take into account the uncertainty in estimating that Emax fixed effect parameter. And while we're doing that, we're going to take all of our other sources of variability, so the between study variability, between treatment arm, and our residual error, and set all of those to 0 to ignore them, because we basically want to consider an infinitely large population and focus on true efficacy. So this, to do this, we're going to do this in Simulx. So I'm going to show you how we would go ahead and set this up. So first of all, this is the project that I've just imported my Monolix project into Simulx. And the great advantage here is I have a lot of things that are automatically set up and created for me. So for example, here under model, I already have my structural model and the statistical model all set up just like I had in my Monolix project. For occasions, I just deleted these because we're not going to consider the between treatment arm variability. For the population parameters, I automatically have imported my different population estimates. So here, for example, my typical estimates are going to give me my estimates for my different parameters with all of my variability already set to 0. So I'll be able to use that. And I have those also, I have my typical and I have my typical uncertain, right? And these are my my typical values, including the uncertainty of this variance covariance matrix. So basically the uncertainty in estimating that parameter. Next, for the covariates, I can create covariates, and this is how I'm going to have my treatment effects, right? I don't have measures of drug concentration. I just have the effect, so I'm just doing the effect with time. And so I can basically create an element with each drug in order to have the treatment effect based on the covariate parameter for that, that drug. And then for output, I can make an output where I have ACR 20. So what this is I'm just doing, I can get directly my different intermediate outputs, and I don't need to worry about the transformation. I can just get directly my ACR 20, and I can say that I want to sample it once a week for 52 weeks. So then if we look at the simulation setup, so to start with, I'm just going to do one single simulation of one typical individual. So we're not yet getting into all of the uncertainty, but we'll just start with simulating a typical individual. I have my output as my ACR 20, and I make 4 different groups that I have each of my 3 treatments and placebo. And for each of those, I make the corresponding covariates that I have the treatment effect. You can see that I also need to have a regressor defined here, but this actually is going to be ignored because I'm not actually taking my between treatment arm variability into account. And so now if I go ahead and look at the output distribution, I have a simulation of a typical individual over 1 year, and I can see the difference between placebo and my 3 different treatments, and I can see, just like when I was looking at my Emax parameter, that after 1 year, I have a lower efficacy for canakinumab compared to my other 2 treatments. So this I can look at visually, but let's say I wanted a more quantitative measure of that. And so for that, I can set up outputs and endpoints. So let me just switch to a project where I've already set that up. So this is everything that in my simulation setup is the same, but now I've added an outcome for this ARC20 last. And what this is this is just the last value, so the value at 52 weeks of my ACR 20. So this is my percentage of individuals that had the 20% improvement. And I have set up a group comparison, which is a direct comparison with the reference group of canakinumab, because I want to see if my treatment is better or worse than these others, or the one that I'm developing here. And I have set the difference as less than 0. So to understand why that is, let's just look here really quickly on the slides. And so what I have here is I have canakinumab as my reference group. and I want to say the difference is less than 0. So the difference is less than 0 means I'm going to compare another treatment, for example, adalimumab, to my canakinumab and take that difference. And so that difference being less than 0 implies that canakinumab is greater than the other treatment, or basically that it's the percentage of patients showing improvement would be greater. So that's the comparison that I want to make. So going back to Simulx. So if I go back to my plots, I basically can now, in my results, I have a group comparison. And so I can see that for this typical individual, just like what I could see on this plot, I can see that I have a positive difference with the placebo, but a negative difference or not success with my 2 other treatments. Okay, so this basically is everything set up that we want, but now we want to take all of that uncertainty into account and actually repeat this over a large number of trials. So for that, I've just changed my simulation. So here I'm doing 100 replicates. So that means I'm going to simulate 100 different clinical trials. And I've also changed here which parameters I'm using. So before I was just using the typical parameters. So it was always just my typical individual. Here I'm using my typical with uncertainty. So I have the estimate of Emax, but also the uncertainty variance covariance matrix, but all of my random effects are still set to 0. So I'm not taking into account between study variability, between treatment arm variability. I'm still using this idea of an infinitely large population, but I'm taking into account the uncertainty that I have in estimating these model parameters. And so what's going to happen is I'm going to do 100 replicates, and for each replicate simulation, I'm going to draw a new set of parameters. So I'm not going to be using the exact same Emax each time. And so what that means is if we go here to the output, now I have an actual output distribution, right? Because I'm not always just doing my same typical individual that you can see here as the median, but I have a whole distribution of possible outcomes. And if I want to look at just the distribution here at the end at time 52, I can look at the distribution here of my last value. So here I can look at that as a box plot, and I can see that I have a wider kind of uncertainty distribution for canakinumab. If you remember, that is the one that was based on only one trial, and it was one trial that didn't also last as long as the others. So I have more uncertainty for that compared to my other 2 treatments that were based on more clinical trials. Then I can go look at the results. And so here I have my 100 different trials that I simulated. And for each trial, I can see if my comparison between groups was successful or not. And remember, successful means that I saw a higher ACR20, so basically more individuals showing 20% improvement for canakinumab compared to the other treatments. If I look at the summary of this, I can see that my success was 100% compared to placebo. So canakinumab would, in all of my trials, is better than placebo, but compared to adalimumab, it's only 43% of the time successful, and only 9% successful for abatacept. So less likely that I'm going to actually have a successful trial compared to the 2 existing treatments already on the market. And so the conclusions that we can take for this is that we saw that the chances are low that canakinumab is going to perform better than the 2 drugs already on the market. As a consequence of this, development of this drug was stopped for rheumatoid arthritis. So this saves the high cost of phase 3 clinical trials. And so this again shows how MBMA can be used to really support really critical decision-making during the drug development process. So this wraps up our second case study, and I'm going to take a little bit of the time here at the end just to go over kind of some of the guidelines of what to think about when you're thinking about how to apply an MBMA with your own data. So first we'll talk about the data set. So we want to have a column that's the study. And so this is going to be representing the study or the publication, which will be tagged as ID, right? Because study in MBMA is analogous to an individual when we're doing a single clinical trial of individual patient data. Next, we have the arm. So this will be the index of the arm that we want to tag as occasion. Any number of arms are possible. They don't have to be the same across studies. The order doesn't matter. It does need to be numeric. But other than that, it can be anything you need. We have time. And then for the observations, so we have kind of our Y, which is basically the untransformed observation. And remember that this is our observation at the study or arm level. So this is going to be something aggregated over an arm, so a percentage, a mean response. But that observation we're going to ignore. And then we're going to need to have another column in our data set where we've transformed that. So this is whatever transformation that we needed to do so that the error model is constant. So we have a constant variance, and that's the column that we want to tag as observation. Then we can have columns for the number of individuals in an arm or the standard error. It's not required to have the standard error. If that's reported by the study, it's good to use it because that avoids kind of that assumption of assuming the standard deviation is the same across studies, of just estimating that single variance for the residual error model. But we can certainly just do the weighting by the number of individuals. And in either case, we want to tag this as a regressor. That way we'll be able to use this column in the structural model. And it's important to note that it's okay for these columns. These can vary by study, by arm, of course, and they can also vary by time. So we could have different standard deviations in the study by time. Also, an arm could vary over time if there's dropouts. Then lastly, we have covariates. And so these are different characteristics that we also might think be explanatory in our model. And this is also sometimes something we can do to control for the heterogeneity across different studies that we can still include in the same analysis. And we need, just like in a individual patient data model where we'd need covariates to be constant over individual occasions, here we need the covariates to be constant over arms. So this is going to be some kind of data aggregated over individuals in that arm. Next, for our model file, this is where we're going to do kind of the building of the parameter. So we're going to separate it into the fixed effects and then the random effects due to between study variability and between treatment arm variability. And so if we're going to be weighting here by an arm, we're going to read that in as a regressor. And that's where we're going to be doing any transformation necessary so that the parameter is going to be normally distributed, because these two are going to be our normally distributed with mean zero and estimated variance parameters. We're going to build the parameter by add them all together. If necessary, back transform. Then we do the part where we calculate our prediction, so our prediction of the response. And then very importantly, we want to transform that prediction to match whatever we've done in our data. So just like that trans Y column we have in our data, we're going to do that same transformation here, whether that's, say, multiplying by the square root of n or dividing by the standard error. And then finally, in the GUI, we want to be able to set up our model in the GUI. And so for our parameter, right, we have it decomposed into these three parts: the fixed effects, and then the random effects due to the between study and between treatment arm variation. For the distribution, For the fixed effect, that just depends on our parameter. It could be normal, log normal, logit, depending on what that parameter represents in our model. But for the two random effects, we want to use normal. And then for the random effects, we want to have no random effects directly on the fixed effects, because we're going to use them here with, for the between study variability, we want the random effects at the study or ID level. And for the between treatment arm variability, we want the random effects at the arm level. For the fixed effects value, for the kind of initial value, we want to just base it on the data for our fixed effects. And then for these two, we want to fix this to be 0, right? Because these are representing normal variables, random variables with mean of 0 and estimated variance. And then finally, for covariates, we can add the covariates onto the fixed effects. If we want to add covariates to our between study variability, we can if the covariates are constant over studies. In the GUI, we can't add covariates to the between treatment arm variability because it's weighted, but we would be able to do that in the structural model if we would like to. And then lastly, I'd like to point out that we have a published tutorial. You can also find this on the MonolixSuite documentation if you look under case studies. And so this is the entire case study 2. So the one of camomumab in rheumatoid arthritis. And so you can download all of the project files if you want to play around with them yourselves. The case study really goes step by step of how to implement everything. And then in the end, it covers some of those same guidelines that I just went over, as well as some specific advice for specific questions. So this really gives you a really good guide or reference for when you want to implement your own MBMA. You can look at that, and this can really, you can use this as really a step-by-step example of how to go through everything that I showed you today. So with that, I would like to thank you very much for being here today.